I'd now like to introduce our second presenter, Dr. Volker Schweikart. Dr. Schweikart is a product application manager for Leica Microsystems. With a background in optics, biophysics, and cell biology, it is his passion to develop instruments and procedures to probe biological processes at the subcellular and tissue levels. At Leica Microsystems, he works with customers, collaboration partners, and in-house R&D to drive innovation in nonlinear optical microscopy and multimodal imaging, including Coherent Raman Scattering Microscopy, multi-photon fluorescence microscopy, lifetime imaging, and harmonic generation microscopy. For a complete biography on Dr. Schweikart, please visit the biography tab at the top of your screen. Welcome, Dr. Schweikart. You may now begin your presentation.
Hello, everyone. Welcome to this webinar on vibrational spectroscopic imaging in cells, tissues, and model organisms using the Leica SP8 CARS SRS system. So I want to give you right at the outset my motivation why one would be interested in using vibrational imaging. So what you see here are 3 examples of images taken with vibrational imaging. All of these samples are on untreated and unstained cells, tissues, and model organisms. And so you can see, even without a need for labeling, you can get very rich contrast, very diverse types of contrast, and you can get dynamics up to video rate with label-free imaging. So the core message here really is vibrational imaging can provide you a very rich chemical contrast. And all of this is possible without a need for labeling, although there can be very interesting applications that actually use vibrational labels, as I will show later. As I already pointed out, quite surprisingly to some people, even though this is a label-free imaging approach, it can be quite fast. And video rate imaging has been demonstrated on live cells, tissues, and even on model organisms. Now, in addition to this rich chemical contrast that you see in the images, vibrational imaging provides yet another dimension on your samples. It provides the entire spectroscopic information on your sample that contains both chemical and biophysical information. In addition to what I just talked, there are exciting new applications of vibrational imaging coming up that make use of vibrational tags that allow you to image targets that were inaccessible by fluorescence in previous approaches, such as small molecules that are simply very hard to visualize using fluorescent approaches. And so overall, I would say this is an entirely new approach to do imaging that is highly complementary and can be very synergistic with fluorescence microscopy. Now, I want to take a complete step back here and begin with the very basics on how it is possible to excite vibrations of molecules using light. And the underlying basis of it all is the spontaneous Raman effect that is illustrated on this slide. So here we are talking about exciting a sample of molecules with laser light, where, as you can see in this energy diagram to the right, the energy of the laser photons is not enough to excite the molecules into an electronic excited state from which they would, for example, emit fluorescence. So that is not the case here. By contrast, this sample will look largely transparent to this laser light, and so all of the light will be essentially transmitted. And only very occasionally it will happen that one of the photons excites one of the molecules to vibrate, and as a consequence, a lower energy photon will be emitted from the sample, where the energy of the photon has been reduced by exactly the amount of vibrational energy that is needed to cause this molecule to vibrate. So in our energy diagram, this process looks like this. The pump photon is being absorbed or used to transfer the molecule to a virtual state, from which it will then spontaneously emit this redshifted photon, leaving the molecule in a vibrating state. Now, this effect is there, but it is a very weak effect. In typical conditions, only 1 in 10 to the 7 photons are scattered in this way. And so therefore, this is a way to do microscopy, but it is a very slow way to do microscopy. And therefore, researchers have been thinking about ways to give this Raman effect a boost. And one of the most exciting proposals and suggestions that came out of these efforts has been the concept of coherent Raman scattering. That is the topic of today's presentation. So in coherent Raman scattering, the idea is to shine in that pump laser that we already used to excite these spontaneous Raman transitions, but in addition to shine in a second laser, which we will call the Stokes laser, where the key is to adjust the frequency difference between the pump laser frequency and the Stokes laser frequency to exactly match one of the vibrational resonances in your sample of molecules. So this condition is called vibrational resonance, and under those conditions, the combined action of both laser fields is suitable to drive the entire ensemble of molecules into a vibrating state. Now, as a consequence, by energy conservation, we are depositing energy into the sample of molecules. Some energy has to be lost from the higher frequency pump beam and is being redistributed into the lower frequency Stokes beam. And those intensity changes as a consequence of the excitation of the molecules to a vibrating state can be measured, and they serve as our image contrast in the process of stimulated Raman scattering. So in an energy diagram, it is depicted here in the right. We are again using the pump laser to transfer the molecules to this virtual state. But instead of waiting for the spontaneous emission of single Stokes photons, we are driving down the entire ensemble of molecules very efficiently by the action of that second laser, the Stokes laser. So again, this is called the stimulated Raman scattering process. Now, this process is most efficiently driven using picosecond laser pulse with fairly high intensities. And under such high intensity conditions, other nonlinear optical processes take place as well. And one of the prominent effects that was discovered very early on was the emission of blue shifted photons, which in an energy diagram can be explained as such, which is called the coherent anti-Stokes Raman scattering process, or CARS process. So what's happening here, the first two steps are exactly identical to the ones in stimulated Raman scattering. However, then the molecules can absorb yet another pump photon to a second virtual state, from which then the blue shifted anti-Stokes photon is emitted. So this is again called the CARS process. And both of these processes are combined under the umbrella term coherent Raman scattering. And they both require, as a key condition, the vibrational resonance of the two exciting laser beams. So both processes only take place when the frequency difference between the two laser fields exactly matches the vibrational resonance of the molecules. So combining those two laser beams into a laser scanning microscope then gives us a way to do vibrational specific microscopy using these coherent Raman processes. Now, one of the key features of this approach is that we can get spectroscopic information. And here in this slide, I want to explain to you how exactly we do this kind of spectroscopy. So the key here is to acquire the images at each frequency difference sequentially. So the numbers depicted here in the upper part of the images, they refer to vibrational frequencies measured in those units of inverse centimeters. And you can see here images taken at increasing vibrational frequencies. And again, each image is taken after tuning the laser frequency difference by one step, and then by another step, and another step. And you can see here that the image contrast that is obtained at all of those vibrational frequencies is quite different, because different vibrational frequencies talk to different vibrational resonances in the molecules. So the sample that I have used here is actually an untreated apple And what you can see in the lower part of the image is actually the peel of the apple. And as you go to the higher parts of the image, we are penetrating deeper and deeper into the flesh of the apple. So what we can now do is we can pick regions of interest as depicted here in color and display the spectra corresponding to those regions of interest. And you can see the sheer wealth of information that is contained in those vibrational spectra that we have just recorded. And from that data set, basically every user can define their own downstream analysis to find out what information is contained in those data. Now, to give you an idea of what can be done, I'm showing you here a so-called spectral unmixing, where we are using those spectral data sets as a basis to discriminate 8 different biochemical components of this sample. And we can then use the information that we find, for example, in the literature on the different biochemical compartments that are present in such an apple to identify or to correspond those different spectral components to different entities within the sample. So here, for example, we have first the outermost layer of the peel, which is called the cuticle proper in an apple. And this phase shows those very highly peaked and very sharp spectral features. Those contain or point to the presence of a solid phase of long-chain saturated fatty acids that are the predominant component within this region. Then as you move just a few microns to the inside of the apple, you will find that the spectra become slightly altered. So you can see in green here, some of those spectral features become broadened. New spectral features appear that were not present in the outermost layer. And this analysis then points to a fluid phase of unsaturated fatty acids. For example, oleic acids would have such a spectrum. Going further inwards, we see those compartmentalized regions with, again, very different spectra. Those spectra point to a very high concentration of polyphenolic compounds, and these actually turn out to be the ones that give the apple their health benefits. And then moving further in, we see spectra resembling polysaccharides such as pectin that are known to make up a major component of the fruit flesh. And then we have, in addition, such more localized pigment structures containing carotenoids such as beta-carotene. So this is just an example of what can be achieved with such a spectral data set. Now here is the instrument with which we at Leica are able to provide all of this information. It is the SP8 CARS system with SRS add-on. So this has been Leica's hands-free standalone solution for vibrational spectroscopy. And so on the left, I'm showing an image of the system. In the front, it is actually the SP8 confocal microscope. And in the back, the blue box is actually the laser source that provides the 2 laser fields that are necessary to excite those vibrational transitions. And so to the right, I am showing a beam diagram where you can see the laser source to the left emitting 2 beams, one tunable between 720 nanometers and 980 nanometers, the second fixed beam at 1032 nanometers. So these are near-infrared wavelengths that are fairly common also in multiphoton imaging microscopy. Those 2 beams are overlapped in space and time and then being focused onto the sample, from which then those coherent Raman signals are being emitted, such as the CARS signals that we can detect in the forward direction, forward CARS, as well as in the backward direction, which we call epi-CARS. And the newest addition to our system, to our portfolio in vibrational spectroscopy, is the addition of a forward stimulated Raman scattering detector that will record SRS signals in the forward direction and thereby complement the coherent the CARS signals. And all of this technology is fully integrated into our visible confocal platform, such that we have visible confocal information available, and in addition, the added modalities of CARS and SRS. And in addition, we can record signals such as second-harmonic generation signals or 2-photon fluorescence with this instrument. So you really have an entire range of modalities available here that you can allow you to generate multimodal images of your sample and therefore maximize the information content that you can gain from your sample. Now, this is a place where a word is in order on the differences between stimulated Raman scattering and CARS, coherent anti-Stokes Raman scattering. You can see here spectra recorded from the same sample at the same time. And you can see that parts of the spectrum are present in both modalities, whereas in other parts of the spectrum, highly rich chemical information is present only in the SRS modality, but is almost hidden in the CARS modality. Now, the conclusion from that kind of data is that SRS provides Raman-like spectra, and in addition, it can be shown mathematically that those spectra can be used for extracting quantitative information, because the signal strength is actually directly proportional to the local concentration of molecules in your laser focus. By contrast, CARS imaging, which was the first modality that was developed, is very great for imaging highly abundant species and can really provide you with the most crisp images of abundant species. However, there's a non-resonant electronic background signal that is the one that you can see here that is fairly featureless in this lower vibrational frequency region. And in addition, the signals display a nonlinear dependence on local concentration, which makes it somewhat more cumbersome to extract quantitative information from CARS images. So that's why there has been a fairly substantial shift of people in the field who used CARS to begin with, who are now all transitioning to the use of stimulated Raman scattering as their preferred imaging modality. And with this background, I want to actually shift towards presenting you a few of the key applications that we have identified for this type of imaging approach. And I want to begin with showing you some examples from the field of cancer research on multicellular model systems. So the sample that I'm going to show you here are cancer spheroids, in this case composed of 3 different types of cells. So we have here a tricellular model for skin cancer that was provided by colleagues here at the Hochschule Mannheim. And what I'm showing you here is a 3-color image where 2 of the colors are chemically specific signals from stimulated Raman scattering. In yellow, a signal representing the total lipid content in your sample, and a image in magenta showing specifically the unsaturated lipids in this sample. And in addition, I'm showing a 3rd signal corresponding to the Second-Harmonic Generation that we can record in parallel, which in this sample comes primarily from the collagen extracellular matrix that is being deposited by those cells. So what can we see here? We can see the 3 different types of cells. The melanoma cells form this outer shell of cells, whereas the keratinocytes and fibroblasts that mark sort of the healthy skin environment for the cancer cells, they are concentrated in the core of the structure here. And the discrimination between fibroblasts and keratinocytes can actually be done, at least coarsely, on the basis of these second harmonic signals, because only fibroblasts are able to produce the collagen that gives this second harmonic contrast. Now, what was very surprising in this sample was the appearance of this highly yellow, therefore highly lipid-rich phenotype of cells. So these fairly sparsely distributed cells here. Those were not expected. The researchers preparing those samples didn't have any idea they existed, but they were very clearly visible at the very first instant when taking those vibrational images. And so, of course, this is a very interesting topic to study what is going on here. So here I'm showing you a more detailed view of this, of the central region of this sample. So we are actually able to take 3-dimensional image stacks using vibrational imaging, remember, we are using multiple photons to excite those signals, and therefore, like in multiphoton microscopy, we get a property called intrinsic optical sectioning, meaning the signals are only generated in the focus of the laser beam. Therefore, by translating the focus of the laser beam into the depth of the sample, we are able to record three-dimensional images of a sample here. Now, what can we do in this sample? So we can use correlations between stimulated Raman and fluorescence imaging to test hypotheses. For example, we just discussed that there's this unexpected force phenotype, very lipid-rich, sparse set of cells. And one of the hypotheses was that they might correspond to infiltrating melanoma cells. And so we used an approach where we labeled the melanoma cells with cell tracker 488 and labeled the keratinocytes with another cell tracker of a different color, and took visible confocal images of those two stains, and then took in addition an SRS image at a vibration signifying the total lipid content. And what I'm showing you here then are those three image channels separated. And you can very clearly see we are seeing green cells, so those are melanoma cells that are infiltrating into the core of the sample. However, those regions where the melanoma cells are found are not enriched in total lipids, as we have seen for this unexpected fourth phenotype. So the conclusion here is that lipid-rich phenotype does not correspond to infiltrating melanoma cells. So we have learned something new, but we are not quite reaching an understanding, and therefore we are continuing research in this direction in a PhD project in combination with the researchers preparing those cells. But what is most important to note for us here is that this example really nicely shows that stimulated Raman scattering microscopy is really feasible even in multicolor fluorescently stained samples, and therefore it shows the potential of using this highly molecular specific fluorescence information together with the more metabolic centered and biochemically driven stimulated Raman contrast to really answer hypotheses on such cellular samples. Now, I mentioned we have spectroscopic contrast available for all of the samples that we are imaging. And so I want to show you a little bit of the kind of information that we have available here. So what I'm showing here is in color. These are spectra of lipid droplets from the three individual cell types that are present, as well as a spectrum of this fourth phenotype of cells. And you can see here those spectra are highly different in some regions, showing that there is a highly diverse biochemical composition of those different lipid droplets from those different cell types. The first peak, starting from the right here at 1740 wave numbers, is a peak that is specific to esterified lipids, and it turns out that those are present in fibroblasts only in significant quantities. So that's a piece of information that could not be possible to record in any other way. Now, the second pair of peaks to the right here, the peak at 1650 and another peak at 1445 wave numbers, the ratio of those peaks has been used in spontaneous Raman scattering spectroscopy to quantify the degree of unsaturation of the lipids present in your samples. So the ratio of the right peak to the left peak gives a measure of the unsaturation of the sample. So we can see here the fibroblasts are actually highly enriched in unsaturated lipids, whereas, for example, the fourth phenotype of cells, which shows this very steep ratio here, is very poor in unsaturated lipids. So we can really clearly identify differences in biochemical composition in a quantitative ratiometric way here. There are many other peaks in the lower energy part of the spectrum that I don't have time to discuss here. There's another very interesting peak down here at about 700 wave numbers that corresponds to cholesterol. And so again, we can see this is only present in the green spectrum corresponding to the fibroblasts. So it turns out the fibroblasts are quite unique in their biochemical or lipid content compared to the other cell types. And overall, what the goal here was to show that there is sufficient information in those spectra to do a label-free discrimination of different cell types in this model system here. And clearly, there's plenty of information to achieve such a separation. Now, what we want to do further is go and understand differences in the response of those cells to pharmaceutical treatment. So I'm showing here on the left an image of untreated spheroids, whereas on the right I'm showing another spheroid treated with a chemotherapeuticum for 48 hours. And you can already see morphological differences here. You can see dividing cells on the left hand. So this is what cancer cells do. They divide as fast as they can. Whereas on the right-hand side, we see a different morphology. We see the nuclei. These are the dark regions in those cells being fragmented, which is a very typical signature of cells going into apoptosis. So we are clearly seeing the cancer cells respond to the chemotherapeuticum here by going into cell death. So that's very encouraging. The morphology already tells us that something is going on with this treatment. But in addition, we have all that spectroscopic information. So we can now really try to understand what is going on with the metabolism of those cells as they undergo treatment. And so I'm showing here spectra of these 3 different cell types. In red, spectra without treatment, and in gray, spectra of cells that have undergone chemotherapy. And we can very clearly see in the top spectrum, the melanoma cells actually show some pronounced differences in the amount of unsettled fatty acids. They also show some increases of peaks at lower wave numbers corresponding to saturated fatty acids. So we think we can see a clear shift of the lipid profile in the melanoma response to the treatment. We also do see smaller changes in the spectra of the keratinocytes and also some minor differences in the fibroblasts, but the majority of the changes is really observed in the melanoma cells as expected. So we really think that this points to the possibility of applying label-free vibrational imaging to treatment, to monitor, to perform drug screening, to understand mode of action, pharmacokinetics and dynamics, and in general, as for preclinical and translational mechanisms. Now with that, I want to shift to the next topic. So the next topic will deal with applications of vibrational imaging in neurodegenerative disease. So here we collaborated with the German Center for Neurodegenerative Diseases, and they supplied us unlabeled mouse brain slices as shown in the right here. So this is a real visible image of those samples. Those are about 100 microns thick slices that have not undergone any labeling procedures. Now what I'm showing you here is a section of this brain shown in a contrast corresponding to the total lipid content in this sample. So what you can see, very bright lipid rich, those correspond to the white matter structures of the brain. You can see on top the cortex region of the brain, which is known to be composed of gray matter, which is more protein rich and less lipid rich. That's why it's bright in contrast. We can even see individual neurons on the left part of the image here projecting into those white matter bundles. We can see those little dark dots that actually correspond to the cell nuclei, to the neuronal cell nuclei. And in addition, we can see, if you have a trained eye, you can see those localized structures here that actually correspond to, as I will show you in the next slides, they correspond to amyloid beta plaques, or to be precise, they correspond to lipids associated with amyloid beta plaques. And those amyloid beta plaques, as you probably know, are at the core of Alzheimer's disease. So this was a sample provided by those researchers that is actually a mouse model of Alzheimer's disease. And so in the next slides, I want to show you what we can do both from an imaging point of view and from a spectroscopic point of view to understand those pathological structures. So here I'm showing you a zoomed in region of this image, focusing on one of those pathological structures. You can actually see there's a shell of lipid-rich structures that actually enclose a core that is dark in this contrast, that is very devoid of lipids. So a very interesting core halo type arrangement of those pathological structures. And what you can see in the surrounding healthy tissue, you can see the individual myelinated neurites projecting through this three-dimensional tissue sample. And what you see in dark, again, these are the neuronal cell bodies and nuclei. And in blue, I'm showing an additional contrast here. So this is two-photon excited autofluorescence that we can observe from this tissue in those localized point-like structures surrounding the cell nuclei. So this additional contrast likely shows a compound called lipofuscin that is known to be present in those brain tissues. So you can see here we have really very, very high spatial resolution images with a very crisp contrast that show both the healthy structures of the brain as well as pathological structures. Now, in a three-dimensional sectioning view, we can very clearly see this core halo type structure that I just indicated. And what we really wanted to use now is our spectroscopic information to understand what is going on in terms of the biochemistry here. So here I'm showing again such a hyperspectral stimulated Raman scattering imaging data set, and I will show you now the spectra corresponding to those different biological structures that are present in the brain. So we go from those pathological structures that are comprised of a lipid-rich halo and a core, and then in addition I'm showing white matter, gray matter, and spectra of the cell nuclei. And you can appreciate how different those biochemical structures look in those spectra, especially in certain regions of those spectra. So in the next slide, I want to focus in on just one of those vibrations, namely the amide-1 vibration associated with proteins, which is actually overlaid onto a CC double bond stretch of the unsaturated lipids in this sample. So zooming in onto this region, the spectra look like this. So we see the healthy parts of the brain, the gray matter, and also the lipid-rich halo structure. They have a peak at about 1660 wave numbers, whereas the core of the pathological structures has a frequency-shifted peak at a different frequency at about 1675 wave numbers. So here are the two corresponding images. And you can very clearly see the most pronounced difference occurs in that core of the pathological structure that is indicated in magenta here. At 1665, this looks void, as in no lipids present in this structure. But at 1675, a very unique contrast shows up corresponding to those fibrillar structures in the core of the pathological region. And these fibrillar structures actually correspond to amyloid beta. This frequency shift is a known signature of misfolding of proteins into a beta sheet configuration, which is known to happen when amyloid beta peptides aggregate into those famous Alzheimer's plaques. So here, by taking the ratio of intensities at those two different vibrational frequencies, we can get a very clear label-free visualization of the amyloid beta content of those pathological structures. And I'm showing a line profile through that structure to very clearly give you an idea of the contrast that we can achieve. Now, this type of finding has been published in two different reports in the recent years, and we have just added our own report on that topic to bioRxiv as well, so feel free to take a look at that story if you're interested. But this was just information coming from one peak in this spectrum, and you can see there's a whole bunch of information more that is contained in the remainder of the spectrum, and I will just focus in on one specific piece of information in the very low end of the spectra here, where the signals are really small, but I want to blow that region up, and you can see that even here in those very low signal regions, we have very specific information here. So there are two peaks that are of interest here, the one at 707 wave numbers corresponding to a vibration of cholesterol, and a second peak just a few wave numbers away, corresponding to various membrane lipids such as sphingomyelin and phosphatidylcholine. Now, taking a look at the images at those two vibrations, we can see the following. We can see at the peak of the cholesterol resonance, most of the signal is actually found in the top of the image, which corresponds to healthy white matter regions of the brain. So those are known to be very rich in cholesterol, which is confirmed by our high image contrast in those regions here. By contrast, the pathological structures don't show a very strong accumulation of cholesterol here. Now, shifting to the second vibration of membrane lipids, we don't see very many of those in the healthy white matter structures at the top of the image, but we do see a significant concentration of those membrane lipids in the surrounding of the pathological structures. So we really have a very clear indication of a differential localization of different types of lipids to healthy and pathological structures in the brain from these data. And so our finding, somewhat surprisingly, was that despite cholesterol being a defining factor in Alzheimer's progression, this cholesterol does not appear to localize to those pathological structures. And so this, again, will now open up a whole new avenue of research on the origin and function of those different types of lipids in Alzheimer's progression. Now, with this, I want to go a little bit quicker on over some of the remaining applications that I'm listing here, just to give you an idea of the potential of our technique in those areas. So here I'm showing you example images of unstained pancreatic organoids. So in red, you're seeing the contrast corresponding to the proteins in those structures. And you can see very clearly the individual cells making up this organoid here. And now in green, I'm showing you the lipids in this structure, very differently localized compared to the proteins. So I'm going to show you the overlay and a zoom in. And you can very clearly see the individual cells being all filled with protein except for the nuclei. And then the lipids being distributed in those very prominent granules. And we actually are convinced that those granules are the precursors to the insulin secretory granules that will be produced by this type of pancreatic cells when they are fully differentiated. However, we were not able to visualize or verify that there is insulin present in those granules, which is very likely from an insufficient degree of differentiation of those cells. So there's a further room for improvement in developing the differentiation protocols further, and then we hope to be able to verify insulin production, which of course then can give us a new, very interesting handle on metabolic disease, digestive disorders, etc. Now, here's an example of that we can actually do live imaging of such organoids. So this is a different type of organoid provided by a different group. And you can see here we have, doesn't play a second time, so you can see the individual lipid droplets within the cells moving, but we can also see contractile motions of the entire organoid. And in addition, we can see here a very highly established epithelial structure, and we can see in certain regions we have those special cells that have a more rich contrast in those vibrational images. Those actually correspond to Paneth cells. Now, another area that vibrational imaging can be really useful is in imaging of model organisms, and I will show you an example of imaging in zebrafish here. So the first image is showing you the protein content in an entire unlabeled intact zebrafish. And I'm going to zoom in now into some special interesting regions. So this is a region, the eye. And you can see here, again, two types of contrast, protein-rich contrast in red and lipid-rich contrast in green, showing you the different layers of the retina at cellular resolution in the intact zebrafish. And you can see here we have lipid-rich regions in the eye, we have more protein-dominated regions, and then in the outer layer, this is where the actual photoreceptors are localized. You can see that there's a even slightly different contrast here. And then the encapsulation of the eye shows up very bright in a different type of contrast, which is likely from second-harmonic generation signals of the proteins that make up this encapsulation. We can look, of course, at different regions in this model organism, and I just want to highlight one. We can see here individual blood cells in the vasculature in the vicinity of the heart. Looking at individual blood cells and even moving blood cells is a possibility using vibrational imaging. Another interesting area that also deals with intact tissues is the area of osteogenesis. So this is a sample provided by researchers that are able to sort of take the skull of the developing mouse embryo and flatten it out and prepare it for imaging. So what we can see here is these very, very interesting multimodal images. So I'm showing you here a combination of visible confocal imaging using M-cherry that in this sample labels the osteoblasts. And then in addition, providing you the nonlinear imaging contrasts, in this case collagen, visualized by second-harmonic generation, and the hydroxyapatite, the mineral that is actually making up the forming bone, visualized by a different vibration using stimulated Raman scattering. And then in addition, lipid-rich structures imaged at 2,850 wave numbers. And you can see in the composite image, all of those different components have a very distinct localization behavior, and we can now begin to understand the interplay between those different types of cells and biochemical structures in the formation of the bone tissue. So this is ongoing research as well. Now, to give you an idea of further application areas, I want to show you a little bit of what we can do in plant biology and food research. Of course, a very well-known model sample or specimen in plant research is Arabidopsis roots, and I'm showing you here yet another example of using label-free vibrational imaging in green combined with fluorescence imaging, both in terms of autofluorescence as well as labeled fluorescence here. And you can very clearly see that we can get three-dimensional information, high-resolution information from those complementary types of contrast, where you can now look at molecular information showing you specific molecules that are labeled with fluorescence, and then the biochemical contrast giving you sort of the context of where all these molecular processes are happening. So we think that this can be a very generic tool for plant and food biology research. Another example that I want to show you here is taking a look at bitter acids in hops cones. So the hops cone contains so-called lupulin glands, the yellow structures in the image on the right here, that actually contain the bitter acids that make up the taste of beer. And so we took a look at those types of structures in this multimodal imaging approach, showing you here SRS imaging combined with various fluorescence colors that we can see from those samples. And in particular, in the lower left of this image, we have one of those lupulin glands, and we can now take spectra of various kinds of or various instances of those glands, and we can see spectroscopically that the biochemical composition can be very different, can be highly varied. These peaks correspond to two known constituents of the bitter acids, and what we think we are seeing here is potentially a signature of the maturation, the degree of maturation of those cones. And so this, in our opinion, will be able to help with analyzing food samples for their degree of freshness, maturation, for their storability, for their durability. So a whole range of applications in food quality and optimizing food production. Another area that is more coming up these days is solid state samples. And here I just want to give you a very brief teaser of what can be done. So this is from a group using one of our instruments, looking at tablets, pharmaceutical tablets using coherent anti-Stokes Raman scattering. And the idea here is to study the crystallization state or the crystal isomorphs that are present in those samples, and also the amorphous state. And all of this information is highly relevant in studies of or verifying the availability of the drug and the solubility of the drug, because it turns out that the preferred state in many pharmaceutical compounds is actually an amorphous state, which will be more amenable to dissolution and distribution in the tissue than some of the crystallized states. And so here the idea was to use different vibrational signatures in addition to second harmonic signals to identify up to 4 or 5 different of the active compound in such tablets, and then to look at those tablets under different storage conditions such as temperature and humidity, and look at the durability of those compounds under those different conditions. So this points to the applicability of label-free imaging for quality control or even for the development of pharmaceutical formulations. So with this, I want to come to the end of my presentation and just summarize up one more time the major areas that we think Coherent Raman and vibrational imaging as in general will have a major impact in the coming years. So we think that preclinical and translational R&D on cancer, digestive disorders, neurodegenerative disease, and also very importantly infectious disease in these days will benefit greatly from the kinds of vibrational biochemical information that we can obtain. The pharma industry is becoming highly interested in those vibrational techniques to monitor compounds both in cells and tissues, to identify localization behavior. Does the drug get to the point where it needs to get to, and what is the mode of action? Where does the drug actually localize and concentrate? But also quality control and development of formulations is possible with vibrational information. Cell and tissue biology will benefit by being able to do metabolic imaging, or an alternative route to metabolic imaging of the fluorescence techniques that can provide metabolic states. We also think that the differentiation of stem cells can be monitored in certain instances, and we think that phenotypic screening applications will come up in the process of those studies as well. Some very interesting applications in neuroscience are being developed. Neurotransmitters can be visualized in certain instances. Even action potential have been visualized in a later way. So there's a whole new area of imaging that is to be developed in the coming years. Plant biology, food biology, biofuels research will certainly benefit, as will the imaging of model organisms for various applications. An area that is just coming up is material science and microfabrication. So here, the opaqueness of some of the samples, option in some of the samples have to be considered, but there are in many cases ways to prepare samples in a way that are amenable to, for example, stimulated Raman scattering imaging. And last but not least, we think that the area of, or the up-and-coming technology of using Raman tags to visualize specific compounds in a vibrational manner can really produce a paradigm shift in optical imaging. And just as a teaser of this, here's an example of a palette of distinguishable vibrational tags that have been developed by Dr. Wei Min at Columbia University, where the sharpness of those vibrational features really allows producing a palette of, I think, up to 40 different dyes that are spectroscopically identifiable and will be able to image, for example, specific organoids in cells or specific compounds by tagging specific compounds. And overall, what I think the vibrational imaging approach really lends itself to is a quantitative approach, where we're going from purely qualitative imaging information to a more quantitative way of looking at data that is, for example, enabled by such ratiometric analyses of spectral features, as I have shown in several places in this presentation. So with that, I would like to thank you for your attention, and I'm happy to take your questions.
Thank you, Dr. Spicard, for your informative presentation. Yes, let's start our live Q&A portion of the webinar. Now, we have lots of questions that have already come in, but to our audience, if you still haven't submitted your question, please click on that Ask a Question box and type in your question and click Send. And we will answer as many of your questions as we have time for. Dr. Spicard, let's get started. Our first question, how does the duration and composition of chemical fixation affect SRS imaging specificity and sensitivity?
Okay, that's a great question. So we haven't explored this in detail yet. What we can say as what we have made our rule is to allow formalin fixation. We have never seen any major effects of formalin fixation on the kinds of SRS signals and coherent Raman signals that we get. We tend to stay away from things like methanol fixation. They tend to remove lipids or affect lipids in the sample. As far as we can tell, formalin fixation is fine and can be used. And this is something that has also been studied in the context of spontaneous Raman spectroscopy. So I think the findings there agree that formalin fixation is certainly something that can be done.
Dr. Spicard, this next question is two parts. How are the Raman spectra taken, and how long does it take to generate a spectrum?
So let me jump out of my current slide and return to the slide that explains this. So the Raman spectra are taken in a quite different fashion from what you would see with classical spontaneous Raman spectroscopy. In spontaneous Raman spectroscopy, you would park a laser beam at one image pixel, and you would take an entire spectrum at that point, and then move your laser beam to the next pixel, acquire a spectrum again, and so on. In our case, it's quite the opposite. So we acquire one image at one vibrational frequency, and then tune the laser beam to another vibrational frequency, take another image, tune the laser again, take yet another image. So we acquire images sequentially. And then after the entire stack of data has been taken, then we can go into individual regions of interest and display the spectra in principle from every image pixel. But the order of operations is different from spontaneous Raman spectroscopy. Now that was the first part. The second part was the duration. How long does it take to acquire? So again, we're talking now about not taking one spectrum, but acquiring an entire, what we call hyperspectral image cube. So the entire spatial information and spectral information. For an example like this one here, it would take about half an hour to an hour typically. So that's the typical time frame that it will take. So something like this is typically best done with fixed samples. With a living sample where things are moving, one would have to be very careful to avoid motion artifacts.
Thank you, Dr. Spicard. Our next question. What is the highest spectral resolution that can be achieved? Could a frequency difference of 5 centimeters to 1 be resolved?
So this is a good question as well. You can see in this example here, we have some very sharp spectral peaks here. Those are about 10 wave numbers wide. And this actually corresponds to the theoretical limit of resolution that should be available in our system, because the duration of the laser pulses imposes a limit on the spectral resolution that is attainable. So about 10 wave numbers is a width of a peak that is available here. Now, can a shift of 5 wave numbers be resolved? Yes, it can. If you acquire a high signal to noise peak, then you can resolve the position of the peak much more accurately than the spectral width of your peak. So I would say the width of peaks that can be resolved is about 10 wave numbers, but small shifts can be observed that are smaller than that.
Dr. Spicard, what objective has been used for imaging the zebrafish sample?
So these, I believe, were 20x IR APO objectives. So these are water immersion objectives with a 20x magnification and numerical aperture of 0.7. But we have a range of different objectives available, ranging from a 10x magnification up to a 40x magnification that we can recommend for coherent Raman scattering microscopy.
I have another two-part question. What dichroic is used in the microscope for the RLD detectors in backward direction? And what is the transmission range?
Okay, so that is a very technical question. I want to answer it from an applicative point of view. So the question probably comes from organizing the way that signals are transmitted to the sample and separating them from the different frequency CARS signals that are being emitted by the sample. So we have two dichroics in our system that we use for different ranges of vibrational frequency. We have one dichroic that is suitable for the wave number range above 2000 wave numbers. So this covers the entire CH stretch region. And we have a second dichroic that is suitable for the wave number range between 2000 and down to 1200 wave numbers. So this is suitable for acquiring fingerprint spectroscopy data. And we have built in our software, we automatically switch between those cubes depending on the vibrational frequency that is chosen by the laser setting. So there's no manual, no need for a manual reconfiguration except for changing external cubes in front of the detectors directly. So that's the question concerning the cubes. And once more, the coherent Raman spectra are limited down to a lower limit of 1200 wave numbers, whereas the stimulated Raman scattering spectra, they are recorded in the transmitted light direction, so there's no such spectral hindrance. And in this case, I'm showing data that go down to 800 vibrational frequency of 800 inverse centimeters, but we can go down to 500 wave numbers with stimulated Raman scattering.
Thank you, Dr. Spicard. Now we have time for one more question, but I do want to reiterate to our audience, and we've had so many questions come in, that those questions we did not answer live today, and those questions that are submitted during our on-demand period will be addressed by our speakers via the email address you provided at the time of registration. Dr. Spicard, our last question for the day. How much average power is required for multi-photon, TPEF, SHG, images acquired with picosecond pulses?
Okay, that's a great question because we get this question a lot. There seems to be a conception that two-photon fluorescence microscopy is only possible with femtosecond pulses. But of course, that is not true. The picosecond pulses, yes, they are longer and therefore they contain a lower peak power, but they are still able to excite such multiphoton fluorescence processes quite efficiently. Not quite as efficiently as femtosecond pulses, but still quite okay. And so it turns out that the powers that we use for coherent Raman scattering microscopy are actually perfectly suitable to excite second-harmonic generation signals and or two-photon fluorescence signals at the same time. So it rarely occurs that we have to change laser settings between acquiring the coherent Raman data and acquiring these additional multiphoton modalities. So these can typically be taken in the same image run.
Thank you again, Dr. Spicard. Do you have any final comments for our audience?
Thank everyone for their attendance. It looks like a lot of people were in the audience. We're really excited to have been able to convey some of our interesting research and applications to you, and hope to hear more questions from you as they come to you.
I would like to once again thank our speakers, Dr. Wayman and Dr. Volker Spicard, for their time today and for their important research. I'd also like to thank the audience for joining us and for their interesting questions. We would also like to thank our sponsor, Leica Microsystems, for underwriting today's educational webcast. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Thank you for joining us, and we hope to see you again soon. Have a great day.